first talk is going to be a remote presentation by Clair Kouba from UC Davis. She's going to give us a nice little update on what's happening with how to predict flows in the Scott River.
Thank you, Randy.
Right. Slide that's great.
Sounds good. And is the audio okay?
Yes.
Great. Thank you, Randy. I think that especially the big picture stuff is just really valuable to keep in mind and keep sort of present as we get into the weeds. And thanks for all the work that you do convening. Today, I would like to talk to this sort of group online and in person about this research that I've been doing, a watershed-specific formula to predict Scott River salmon outcomes using river flow metrics. My name is Clair Kouba. I'm a postdoctoral researcher at UC Davis, but as early as July 1st, my contact information will change. I'm taking a new postdoc at Yale, which is why I'm presenting to you guys from Connecticut. Another way to rephrase this title is which flows are important for fish outcomes and what does the data say? And I want everyone to bear with me for the next dozen or so slides. I realize that everyone in this gathering thinks long and hard about these questions in great detail. And I'm here to sort of describe this radically simple approach that we took in the research that I was doing for my dissertation at UC Davis for the past 6 years. So I will just briefly talk about the objective of this research project, the proposed approach, and some preliminary findings. Disclaimer, these are still preliminary, and during the peer review process, they're subject to change. We are trying to get this published in the immediate near term. So what are our objectives for this project? Let's start with some really simple questions. How does management, we think of management as a screwdriver and a wrench, change flow? And how does a change in flow affect these ecosystems? For this first question, we have a fair amount of tools at our disposal, and these include things like a well pumping test. You can run a well really hard, you can measure to see if it affects the flow in a river, and that can inform your information about that specific well, that level of management. There are pilot projects going on in the Scott Valley where people are building beaver dam analogs or running managed aquifer recharge projects and monitoring the effects that that has on flow. You also have tools like hydrologic models, like the model that we work with at UC Davis at the basin scale that's connected groundwater and surface water. And you can use that to ask these big picture questions like if we have radical changes in the weather or in water use or land use, how would that affect the flow in the surface water system? So we have a lot of tools to look into this first question. And I would argue that the second question, in spite of decades of work by lots of people here, is still a harder problem. How does the flow affect the fish? So if we zero in on that, we took the opportunity to ask, can we express this with numbers? What is the quantitative relationship here, and what units should we use? And if we sort of back up and think about all three of these, in terms of units, we have several options for management. Management can be thought of in terms of, for example, acre feet of water applied as irrigation, applied as managed aquifer recharge, acre feet of water purchased in a water lease. You can think of management in terms of acres of crop coverage or acres under a certain type of irrigation technology. You can think of management in terms of dollars invested in a construction project or a dam removal project. So management has a lot of quantifiable units that we can measure. Flow has, because you can slice and dice a hydrograph in all manner of different ways, there's almost an infinite number of units you could use to talk about the flow. Some that come up a lot in Scott Valley management discussions include the dry season minimum flows, the timing of the fall flows that are needed for salmon access to reach their spawning grounds, the duration of the wet season, and maybe the timing of the spring flow recession as the snow melts. So all of those units are available. We just have to process the flow records that are continuously monitoring it to calculate those. Lastly, we have fish numbers, and this is really the rate limiting step here. There's been a huge investment in Scott Valley, and as we saw in the maps that Randy was presenting, in the Klamath Basin writ large. For this ecological monitoring, it's labor intensive and expensive, But because of all these efforts, we have data on number of spawners. We've done red surveys. We have number of eggs laid. People have estimated the number of juveniles. So we have this time series of ecological data. But in spite of this abundance, it's still less than the other two categories. So this is sort of where our predictive power is this is the make or break point. So we'll focus on the size of the ecological data set as one limiting factor in this predictive exercise. So our proposal is we would like to seek units and coefficients to boil these two relationships down into very simple math. And you can call this, if we have a change in management, if I'm planning to add this many acre feet of water as a managed aquifer recharge project, can we find a coefficient that is flow over management units that turns that number into, this is the change in flow we expect to see in terms of the timing of the fall flows or the base flow magnitude. Similarly, if we have a change in flow in whatever units we're considering, is there a magical coefficient that can convert that into a change in a quantitative fish outcome, like the population reproduction? These are of course limited to freshwater system experiences for the fish, and as we know, there are many other factors that are not flow determining these outcomes. So this was sort of an exercise in seeing how far could we get in explaining some of these ecological responses using just the flow data. This assumes a linear relationship, and in case anyone wants the math refresher, this delta we're using is this triangle symbol, and it means change in this. So we're talking about perturbations to a system in terms of management, changes in flow. And if we cancel these units, we could theoretically achieve some sort of ultimate forecasting tool where if you had a planned management action that you wanted to take, you could convert it into a potentially forecasted impact on a quantitative fish outcome. So that would be ideal. Now, that's a lot. That's a giant project. In my work, I did my first dissertation chapter on this second link, where we're trying to find the delta fish delta flow equation. And I did my second chapter focused more on the first link. And for the rest of this talk, I'm only going to talk about the second part of this, but I did want to put this in context for this is our full motivation for why we think this is valuable as an exercise. It would be great to be able to take this fish flow relationship and apply it to some of those other projects. So what's our proposed approach? Just for some background, a classical prediction method is least squares regression. And most people have looked at graphs of this. You take a cloud of points and you draw a line through it, and the slope of the line and intercept that minimizes the distance from all the points to that line is that slope is your coefficient. You can assume a linear relationship between one or multiple predictors and your outcome, and your predictive power can be estimated with things like correlation coefficients. This classical method struggles with overfitting if you have a lot of predictors or not very many observations, which is the case in this ecological data set where we're limited by our ecological data. And just a visual example here is a strong correlation where, or a stronger one relative to this other one where the dots are closer to the line, and then there's a weaker correlation where there's a lot more scattered. Now, instead of that, we're proposing an approach with several steps, and I'm not even going to talk about all 5 of them. We're just going to talk about the 2 central ones, which is for these 2 species of concern, coho and Chinook, can we select the most predictable ecological metric based on all the data that's been collected, and then estimate those coefficients of our hydrologic metrics using lasso regression, which is a spin on least squares regression. So let's talk about the ecological metrics first. On the graph here, we have our ecological data categories on the top. We have the number of Chinook spawners, number of Chinook juveniles, the Chinook juveniles per adult, sort of a normalized metric. For coho, we have some more data. We have the number of spawners, the SPF or smolts per female metric, the juvenile abundance estimate, the percent smolt survival, and the number of reds. So all of these different numbers are time series of ecological data that have been collected and are available as a response metric, but like which of them is most related to flow was one question. We have an example predictor. For example, the minimum flow in the 2018 dry season was 6 cubic feet per second at the Fort Jones gauge. And an example response, if we assumed they were related, is that the spawner abundance that year was 739 individuals. And you can take time series of these predictors and responses and calculate the correlation coefficient between all of these pairs to estimate how related they are. So you can visualize that with these blue and red dots. The larger the dot, the stronger the coefficient. Things are negatively or positively related in red and blue. So boil it down, which did we pick? We picked two normalized metrics, the juveniles per adult and the percent smolt survival that's based on current methodology. It is still sensitive to things like new data points. So what is lasso regression, our proposed method for making this prediction? It's an acronym. It means least absolute shrinkage and selection operator. It is a method of predictor selection. So if you have a huge number of hydrologic metrics, you can narrow down your best candidates for making a predictive model with a slope and an intercept, or multiple slopes in this case. This is a less flexible method than standard least squares regression. So this helps reduce the risk of overfitting, and it's good for situations where the number of predictors can exceed or approach the number of observations. It minimizes this quantity, which we don't have to get into, but this value lambda is called a shrinkage penalty. So it basically penalizes more complexity in the model, and selecting your lambda value means like saying, I want a simpler or a more complex model. And I will, if it's more complex, I will take the risk of like overfitting and adding, random noise into this predictive model. Our preliminary findings can be sort of summarized in these three graphs. On top, we have the regression coefficients, and they're all standardized so that you can see that as you change your lambda value on the x-axis, higher values of lambda mean that there's like zero coefficients. This means we're just selecting the average value of the response. That's our best predictor. And we're not going to incorporate any information from hydrologic predictors. As you reduce the lambda value, you start to add in more and more complexity to your model. So if you wanted a one predictor model, you could sort of find yourself on the red line. The one predictor it would identify for you, it would select, is the rearing year dry season magnitude 90th flow percentile. That's the one that it has picked out of our huge bag of hydrologic predictors. And on the second graph, we can see that this explains up to about 20% of the variation in the ecological response. That's not a lot. That means there's a lot else going on that's not explained. If you wanted a three predictor model, you could find yourself on the yellow dashed line. And the two extra predictors it would add in to the mix is the log of the sort of fall flow volume in the brood year, the period when the eggs were laid for that cohort of salmon. And secondly, the timing of the fall flows when the flows rose past 70 cubic feet per second at Fort Jones. And this three predictor model would explain up to about 45% of the variation. And I want to point out in the final graph here, the third one, as you add more and more coefficients, if you perform a split the data in half, create a new model and test it on the other half, you can see that test error becomes significantly larger as you add in more complexity, because you are at risk of adding more random noise instead of adding meaningful information from the hydrology. So we want to find selecting the lambda value was an exercise in balancing, well, we do want to incorporate maybe more information from the hydrologic predictors, but we don't want to risk, it's hard to know at what point that's overbalanced with the random noise that you're introducing. So we've selected a value for the purpose of this talk, but that's still an ongoing discussion. For Chinook, similarly, the top predictor was that log of the flow volume in the brooding year when that cohort of salmon was eggs being laid, and then explains up to 8% of the variation in ecological response. For a three predictor model, it explains up to about 15%. Now, we can take these predictive models for coho and Chinook, and because we have hydrology for all of the years dating back to 1942, from the Fort Jones gauge, we can predict how, what percent of smolt of coho survived and what the fraction of juveniles per adults was for Chinook for all of those years. The gold triangles on these plots are the observed data, the ecological data that we have. The black dots are the predicted values for all those other years, as well as the ones with observations. And then the gold bars are our error term. So that's the difference between predicted minus observed, and that's telling us how effective the model is. Also, there are some red diamonds on the Chinook plot, because in this model, it is possible to come up with negative values, which are physically implausible. There is no consumption of juvenile Chinook taking place due to the hydrology, but the we wanted to keep those in here as a sort of visual representation of uncertainty in this exercise. And I would say, based on these results, we have had some success at predicting whether an outcome in a given year would be high versus low, but it seems to be a lot worse at predicting high values. So I would say this is not capturing all the relevant information that the fish populations are responding to when they have a good year. So what are the differences between the two species? Coho variability can be more coho variability can be explained with a smaller number of variables. And I think there's a couple ways to interpret that. One is that possibly Chinook outcomes are just less predictable than coho, full stop, but it's also possible that they're less limited by the hydrology, and so they're less sensitive to it. And maybe they are more sensitive to other factors like water quality or interior population dynamics that are not captured in these flow metrics. A disclaimer at the end here, with this small of an ecological data set, these findings are highly sensitive to new observations. So as we gain more years of observation data, I'm sure some things in here will change, but this approach can be replicated every time we get new information, and we can check the robustness of some of these findings to see which hydrologic metrics continue to crop up as like valuable predictors. But we're nevertheless excited about this research because it would allow us to take hydrologic model outputs, such as the scenarios that we developed for the groundwater sustainability plan in Scott Valley. We can analyze the resulting changes in flow in terms of these metrics, and then use it to estimate or predict a fish impact for the whole model time period based on that management action. Thank you. I'm happy to take any questions, or feel free to contact me after the fact.
All right. Thanks, Claire.